So let's see. Yeah, thanks a lot. Actually, first of all, I'd like to thank the organizer for inviting me, and of course also for organizing this short but really interesting symposium. I like the title. The organizer asked me to say a few general words about ecological immunology, which I'm happy to do, feel quite honored. And by necessity, of course, it's not possible to do justice to the whole subject in 20 plus minutes, nor will I be very balanced. I'd like to make some points, and some of you will have that heard before or heard from other sources, but I still hope there's some elements in there which will be new and interesting for you. So having said that, if I'm correct, we press that one. End of slideshow. That is a very good start. Okay. Actually, my title, it is without a hyphen. And the field took its name, as we think all agree, by and large, with this paper by Sheldon and Verhulst, who ironically at the time didn't work on in this area at all. But essentially the argument was very simple, that immune defenses or defense against parasites are necessary because there's so many parasites around and they have evolved. They're adaptive, but they're also costly. And this is by and large, or was the agenda, still is the agenda of behavioral ecology. And if you go through the paper, of course, there were 3 topics mentioned. One is about the costs. What are the costs, in fact, of defenses? Not purely in calories, but actually in sort of losses of other opportunities. What is the effect on sexual selection, which went back to the famous paper by Hamilton Sook about bright birds and parasites. And finally, I must say a little bit of a lip service, what is the consequence of immune defenses for population dynamics? Now, jumping into the subject, here's an example of costs. That's a typical kind of work that has been done over the last, let's say, 20 years or so. And it shows, this is an example from our own work, yes, immune response has a cost. In this particular case, it's a social insect, so the bumblebees, which we work with, and they live in colonies, obviously. And what we did in this particular example is to have one half of the colonies challenged, immune challenged, so that the immune system is activated, but actually they do not get sick by a parasite. They just have to use the immune system, and the other half is the control. And as you can see from the figures, it's a very clear effect that if you have to run the immune system, actually you have less fitness, which in this case is measured by the production of new queens, young queens and males, which are their reproductives and go on into the future. Here the controls have less of an effect, or is the reference, and as you can see here in this example, yes, there is a cost at the level of the colony if you challenge the individuals constantly, as is shown in the image here. Now, what happens if you go to the field? So we repeated this experiment in the same sense. Here you can see a co-worker basically attending a colony in the field here, and half of the colonies were challenged, the other half was not, and the question was the same again: would this lead to a cost as postulated or original agenda of immune defenses? And as you can see, there's a clear difference between this group and this group, but unfortunately in this case the difference is not actually traced back to whether the colony is immune challenged, has to run the immune system, or whether it's a control. But actually the difference here goes back to whether you add a little bit of food or not. So in the field, the whole difference, which was highly significant in the lab, vanished into insignificance, and there was no tangible effect. So this shows us, of course, that what we see in the lab, which is no news, may not necessarily be the case in the field. Now, of course, this leads us to the other end, which is the other branch that has started to come into Eco-immunology, which I think originally is certainly a branch that originates from considerations of population ecology. And it goes back to at least people like Charles Elton, who started with what you always have to start with, namely looking for patterns. So how many parasites are there? How are they distributed? And in this particular case, it's the number of worms per mice distributed over the year, over years and over the season of the year, so first, second, third quarter of the year and so on. And in more modern times, of course, you see the same kind of spirit in the analysis of the classical study here by Pete Hudson of the red grouse, that the more worms a bird has on average, the lower actually the growth rate of the population, and the higher the mortality during breeding, or the lower actually the fecundity. So, of course, this is in the same spirit, and actually goes on, and I cite gladly some examples from the work done here in Liverpool, which is in the Banqual and cowpox, essentially the main system. And you can see that actually, for example, if you look whether an animal is infected or not, this has an effect, a slight effect actually on the age of maturity. So infected ones tend to reproduce at a later age than non-infected ones. It gets a little bit more murky if you look at the effect actually on survival. Sometimes the effect on survival, according to months here, is better survival, sometimes worse. Most of the time, there's not much difference. So what we see here is not very different from what we've seen in the bumblebee example, namely that sometimes it's very difficult to see effects. And actually, if you look closer, of course, you have lots of other things going on, like co-infection with other parasites, and so on and so on. So wouldn't it be nice to do direct test? Yes, coming back to this classical example, yes, you can do so. Here in this case, if you compare the first with the last panel here, this is the control situation. The population cycles over the years in abundance. And if you remove the parasites by treatment, the cycles collapse. Wonderful example of doing a field experiment. But it's not immunology. So how about immunology? Well, this is another long-term classical study in the soy sheep, done by Andrea Graham and others in this particular case. And this is actually the result of lots of statistical analysis here, fitted data, and so on over many, many years. And as you can see, strangely enough, as you increase the titer of ANAs, which are anti-nuclear antibodies, so antibodies working inside the cell nucleus, actually survival gets better, but only in winters with harsh conditions, crash winters, not so much in non-crash winters. But here we have some traces of linking the immunology back to the field situation. Don't ask me why it's particularly ANAs. Maybe Andrea can answer this question. So we have these two branches, one originating obviously from behavioral ecology, the other one from ecology and population biology. And in a way, not surprising, we have different kinds of questions. For example, the left-hand branch is basically interested in things like adaptation, what explains behavior, whereas the right-hand side, I think originally and still is, mainly actually concerned with what controls populations and how important are parasites for that. And it's no surprise to see that the questions then also differ. For example, here it's about trade-offs and costs, the consequences, the selective value of immune defenses, for example. Here it's more things like understanding epidemics, reasons for cycles or population sizes. And you get in consequence, you get these kind of questions, which defense means here essentially it's immunology, interest in immunology, where here it's more actually what it means for natural populations. Now we come back to that a little bit later. It's fair to ask, what have we learned from the behavioral ecology branch, the left-hand side branch? Now, I'm shortening it, of course. It's not doing justice to the whole breadth of the subject. Yes, we have learned a lot about costs. Here's a table, and it goes on and on and on. These are just energetic costs. So people have shown, yes, indeed, using the immune system is energetically costly, and it has some consequences. You can't use this energy for something else. You can go on, and people have done that. Yes, of course, it is true that the cost depends on ecological parameters, such as, for example, nutrition, body condition, such as season, temperature, local population density, and so forth. Yes, we have learned a lot about the costs, which is the original agenda. So costs are real, but are not always visible. Sometimes they're even compensated. They depend on the environment. They can be of many forms, energetic, fecundity, life history cost, and so on. They can associate with different elements of disease, of the defense, and so forth. So I guess we can summarize, yes, so far, the ideas are basically correct, but from this branch, it's not quite clear how it transports back into the field. So amazingly, as we went along, and I'm coming back to this point again in a while, we know more and more about the immunological side, so actual immunology, and I think it's interesting to compare that to the other branch here. What have you learned from the population ecology branch? Well, a lot as well. For example, that the importance of epidemic phenomena, of life history problems, seasonal variation, dynamics of populations in general, of course. In the wild, there's a problem with having different parasites. Sometimes the host behavior changes, so you don't see an effect. Sometimes it's also a straightforward problem of measurements. Is it the right measurement or not? Nevertheless, sometimes you get the luck to have some immune correlates, like antibody titers, the example I just showed you. But alas, I say by and large, the effects are sometimes not very clear and often weak. Of course, they're good examples where it works, but it's also tough terrain. So we can probably say, yes, the ideas are basically correct, but sometimes they're not lost in translation, but really in complexity. And somehow that shouldn't surprise us. So if you make a short summary at this point, I think ecological immunology, without hyphen here, has had its successes in both branches, obviously. It's probably closer to immunology when it comes from the behavioral ecology side. It has allowed us to get some insights into the kinds and signatures of selection, obviously, on the immune system. I think particularly in the behavioral ecology branch, where many people have worked on insects so far, there has been a lot of emphasis in unraveling the immune mechanisms. And presumably this is an insect problem, because we know so little about the insect immune system compared to the vertebrate immune system, that people have gone into that and actually tried to clarify the immune pathways in particular. My feeling is we should not go too far into that. Actually, remember that this is this famous phenotypic gambit that Alan Grafen suggested. Let the mechanisms to some degree be mechanisms and see what actually the in and output relationships are. That would lead us a little bit further, I think, in many areas. But by and large, I think we have major deficits, and again, I think that's not very surprising to say that, to putting ecological immunology back to the field. Now, if you have difficulties, sometimes in animal behavior, there are displacement activities. You do something else which is successful and you get a lot of money. So these are things to the rescue. Now, I don't mean disrespectful, please, or actually sort of disqualifying. I'm just trying to make the point here. So what are these rescue exercises? Well, I think one is genetics and genomics, and I don't want to exempt ourselves either. You don't have to understand this graph in particular. It's an insect cell, comes out of the Bombus Immune Genome Project, and it shows the different immune pathways. So generally you can sort of distinguish a recognition section, so where the parasite is recognized, let's say outside the cell. There's a signaling compartment where actually the signal is transported, and at the end of the day, something is produced, an effector that kills the parasite, for example. Now looking at that, wouldn't it be nice to sort of shortcut our problem by looking at what kind of signatures of selection we have in the system? That would actually add something to the understanding. Yes, it can do that. Let's have a brief look here. Here are examples. This is now from the insect world. Traces of selection on immune genes. Now we can look at the actual sequences, so the series of nucleotides. And what you find is, yes, indeed, there is often faster evolution on immune genes than non-immune genes. There's typically more positive selection to be found. Interestingly, purifying selection is not as common as you might actually expect, so people have difficulty to find that. If you look at the compartments in the recognition genes, so out there, yes, there is selection, but it's quite inconsistent, so it depends a lot on what kind of receptors you're looking at. If you look at the signaling genes, so what's happening inside the cell, yes, positive selection, sometimes purifying, you find that. If you look at the compartment of the effectors, for example, the antimicrobial peptides, which occur just everywhere, surprisingly, they're mostly conserved, with few exceptions. But you get an antiviral pathway where you have strong signals of something happening. So yes, there is evidence for positive or purifying selection, but often it is surprisingly weak or absent. This is actually perhaps a problem of looking at the canonical genes, the ones we know, not the ones we don't know. They're somewhere in the genome, but they haven't yet been identified. Perhaps that's one of the answers. There's a lot of happening at the level of gene families. So typically, genes duplicate, and after duplication, the duplicate assumes a new function, so you get entire families. And if you look at that, you can see a quite clear difference, for example, between signaling genes, which are typically single copy orthologs. In the recognition, so this is inside the cell, because they have to work together tightly, presumably it's controlled in that way. In the recognition section, you have common expansions of genes. You also have actually lineage-specific expansion, so you have completely different genes in this lineage than in this lineage. And in the effector family, it's quite similar to the recognition genes. We have differences in, for example, the kind of AMPs, and so on and so on. So to cut it short, yes, there is evidence for evolution of immune genes at the family level, but typically, as I say, entire families change, particularly in the recognition, the effector compartments. Whereas the signaling part seems to be rather conserved as a functioning machinery. So we get a little bit of insight. We get a little bit of insight, but in fact, it is also not as clear sometimes as one might wish. Now, of course, it would be a little bit naive to expect that everything is clear. but that's where we are. So in a way, we can also ask why it is so difficult to find these correlates, genetic correlates, for example. Maybe it's not research enough, that's an easy one. Maybe the preconditions for adaptations are not there, so actually the necessary genetic correlations are not actually around. Maybe we don't know which parasites we have to look for, so maybe multiple ones, so you get a lot of compromise, so to speak, in the immune system. Maybe it's also a very simple answer in the sense that the immune defense is already very complex and plastic. That must limit to some extent the traces of selection as you go along. And finally, it's also quite possible that there are more heavy internal constraints. The system has to work together rather than external ones. So all of that, of course, is possible. Well, I come to the microbiota in a second. Another rescue system, of course, is rather than to study tedious ecology, you look at the mechanisms, which I was already mentioning. So genomics, of course, is something. Looking into immune pathways is, of course, another consequence. This is another way to study mechanism, which is an example of our own lab. Here the question was whether the antimicrobial peptides would be effective against these parasites we're studying, which is a trypanosome here, Critidia bombi. And they are effective, and actually they're very effective also in combinations. So we were asking ourselves, are particular combinations of concentrations of these peptides particularly effective against this parasite? So here you have the space of effects. This is concentration of one, this concentration of the other. And you can see in this landscape, this is the best effect. Actually, this is the best effect for this strain, genotype, and the effect, the best point is different for another strain. So yes, there are, of course, nice mechanisms to study, and they keep us very busy, and they're also relevant because in this particular case, it's about interaction between host and parasite genotypes or backgrounds, and whether or not there's some specificity mediated by that. But it doesn't answer yet the question how this transports to the field. Not at all. Theory is always good. Here are two good examples, but frankly, I must confess there's a lot of theory out there which is quite useless, to be honest. But there's some good examples. Here is a good example, trying to find out what is the optimal investment in immune defense, or trying, for example, to see how the dynamics of populations emerge from parameters of the host-parasite interaction. If you go to the left one, that's actually a good question. Can we identify the best defense? I mean, that's a very tall order. And here is one example which comes close, which is from the Stickleback study of the Walensky and co. Workers. Here you have actually different allelic variants or different number of alleles in the MHC locus. And as you can see here, the question was whether this relates to survival of the fish in the natural environment. And in this particular case, yes, it seems to be an optimum function in the sense that an intermediate number provides the best protection. So that's the kind of question which is really challenging, but very interesting to follow up. Can we identify a best defense? Finally, microbiota always reminds me a little bit about the Greek drama. You know, if you have an impossible plot and want to resolve it, there's a deus ex machina coming down from above and rescues your plot. Sometimes microbiota is used like that. We also have studied microbiota, so I'm guilty myself. Here's an example of the latest one, and of course they're interesting results. The question was, for example, can we predict how protective a microbiota is against our parasite by simply looking at some ecological parameters? In this particular case, the species or taxonomic diversity of microbiota. And as you can see, there is a correlation here. In fact, it goes the opposite from what you would naively think, which in this case is the more complex or diverse the microbiota is, the less protective it is. You can discuss this if you want, but that's the finding. Yes, microbiota, wonderful, and it can do a lot of genomics and genetics, and it's actually helping to some degree. So, if you look at these two branches and what I just said, what is actually happening in this interaction, or what's happening in this interesting space between areas? And it's actually quite interesting to note that this branch here is more or less vertebrates. Actually, it's more or less mice, or a large thing is mice and mammals, I should say, soy sheep. I'm not sure whether there's any good study in insects that actually covers this disorder. Here are many insects, because they have many advantages to do experiments, obviously, in the lab, and they're also easy to keep and so on. So there's also a taxonomic divide here, which is sometimes a little bit unfortunate actually for the discussion. But what is happening here? Well, if you look at that, maybe we should reflect back to the beginning. What was actually the meaning of ecological immunology? And I think, to put it short, here, the idea was that immune defense is an adaptive trait, which is subject to constraints, costs, and trade-offs, something like that. Now, if you do that, then immediately you come up with quite a long list of questions. For example, which adaptation are we actually talking about? Can we, for example, identify a key parasite where the adaptation goes to? Or is this impossible? We tend to have, for good reasons, systems where we can easily work with. Are there correlates with environment? I'm trying to mention a few things. Are there, for example, we can also ask, are defenses more variable or more diverse, for example, in a complex habitat? Or is the plastic adaptive immune response, so the one that is plastic, more common in unpredictable environments than not? No idea. Does the immune architecture, for example, the set of antimicrobial peptides correlate with the habitat? We recently tried to do that, and the question mark remains. There's almost no study that actually has addressed that, and the few that are have no conclusive answer. So these are very basic questions, I think, which are still there. Is there a convergent evolution of the immune system in different habitats? On the other side, I don't think we should shy away from asking stupid questions, which is actually parasite pressure more important than physical chemical conditions in a particular situation? Is immune defense important at all? Of course it is, but you have to show it and find out. Are the costs relevant, or are they just a live artifact, or sometimes popping up and disappearing? Of course, I think it is, but we need data, and of course, I know many of you have done that and have found good results, obviously. So this also means, of course, that you go beyond correlations. The tests will be quite challenging, actually do that. Constraints, actually a very nice example is the vertebrate versus the insect immune defenses. So insect immune defense have only this innate immune system. The vertebrate have the adaptive one in addition. I doubt whether there is actually a consequence of that for population dynamics. So the immune system, how it is structured, has probably no effect on the population dynamics. It may sound trivial, but it tells you something, that namely there's something beyond mechanisms that one has to take into account, obviously. Quantitative predictions, like good old days of optimal foraging. Are we able to predict how many antibodies should be produced in response? Quantitatively, maybe possible. And is ecological immunology actually good for a problem, to solve a particular disease problem? So the flagship has been quite successful. I don't think it's going to sink here, I'll show you here. But there's still a lot of white areas on the landscape, obviously, and I think it might be good to think back what actually the agenda was. Not that we should slavishly follow that, but just to reconsider what actually our interests are in the first place. And if that is successful, I think there will be continued a new life for an already successful discipline, but which is sometimes, I think, in danger of losing itself too much into things that are easy to finance, but actually may not add so much to the agenda as we might hope for. So with that, thank you very much for the attention.
Fantastic, really nice introduction. Have we got any questions or comments? Please wait for Rosie to come out of the microphone.
All exhausted.
So I guess you said the stupid question, is immunity important? And then you said, of course it is. I guess I'd ask the stupid question again, is immunity important in terms of how much variation in, say, infection levels or disease in a natural system is explained by variation in the immune system?
That's exactly the question in quantitative terms. I mean, we should have a better understanding how much of the variation we see is actually due to the differences in immune defense mechanisms. Do you know? I have no figure. I have no figure. But I think we wouldn't work on that if we were not convinced it is there. It is a significant proportion, but it may make a difference whether it's 10% or 50%, obviously. Does anybody have an answer to that question? Well, it would be the basic question. Yeah, that, I think nobody doubts that, but I recall Lars' study, for example, when he shut down the adaptive immune system in mice, wasn't it? It was an experiment, obviously, but not much happened, essentially, right? So it seemed to be a way dispensable. Of course, we would be crazy to believe it has no function, obviously, but yeah. But no, even with the genetic signature, you know, we think that's very clear. You just go in there and you find it. Yeah, sometimes not. So it's not as straightforward, and I think this makes it both interesting and frustrating, of course.
I guess the question what if you use 10% less? That's what we want, which is 10%.
Yeah. Yeah.
It's a better question.
Yeah, maybe. We'll soon know somewhere.
Rising challenge of why immunology in this context is important, and I think it's because it's kind of integrated. I think a lot of where population ecology came from here was the question that comes back to Elton as to what role do parasites and pathogens play in population dynamics, and there are so many of them.
Yes.
So many of them that we can't see. I think one of the arguments for studying immunology is that you don't have to bother about the individual parasites so much. It all becomes integrated into the host response, the immunological response, and it's kind of a very, very useful policy way of studying all the parasites and all the pathogens.
Yeah, I quite agree. And I think what's certainly also the case is that all these infections presumably always, or most of the time, they're controlled by the immune system, and you just don't see any effects. And the immune system is just busy adding a little bit of load, and of course, when you start to strain the system, then the whole thing explodes, obviously. So that's certainly probably the normal case, which makes the study a bit difficult sometimes, particularly in the field. But that's the order. But I absolutely agree, and I think the focusing on immunology, of course, is correct. But I'm just saying sometimes, and just particularly from the world I come from, I think sometimes people have gone too far into the details, and there are riddle immunologists which honors them, but it should not be sort of our job, so to speak, to actually unravel the pathways of immune defenses. It's actually a question of how much we should know about it, and that's not an easy question to answer as well, I guess.
Okay, I think we should move on, but I'm sure these bits will come up again and again through various talks next talk are really nice things. So no further ado, should we move on to the second speaker? Lars rather than the intro.
So how do I get out of your? Ah, here we are. Okay. So first of all, thanks to Andy and Mike for giving me the opportunity to present here. So today I will talk about how hosts defend themselves against an infection. And I will first go through some concepts and then show some results from an infection experiment with gut nematodes in lab mice. So the aim of host defense is to reduce the virulence of an infection, and that can be done in two different ways. So to illustrate this, I will use this simple graphical model where we have host health or fitness on the y-axis and then infection intensity on the x-axis, and where infection intensity is the number of parasites or pathogens per unit host tissue. And it seems likely that the host health somehow declines with increasing infection intensity, and this function can take any shape, but for simplicity we will now assume that it's a linear like this. So virulence then is often defined as the effect of an infection on host fitness or health. So in an infection with this level of infection intensity, the virulence is simply the difference between fitness or health in the uninfected state and fitness at this level of infection intensity. So to reduce this virulence, hosts can either increase their resistance, where resistance is a host defense that limits the replication of the parasite, so it reduces the infection intensity and thereby also reduces the virulence of the infection. Alternatively, hosts can limit the virulence of an infection by increasing their tolerance, which means that they reduce the slope of the relationship between health and infection intensity. So tolerance is a type of defense, a type of host defense that reduces the virulence by reducing the effect of a given infection intensity. And before we go on, I would like to point out that both resistance and tolerance are host traits. So when we talk about resistance, we mean that there is variation among host types, for example, different genotypes, in their ability to control the replication of a given parasite. Similarly, when we talk about tolerance, we mean that there is variation among host types in their ability to limit the damage of a given infection intensity. The parasite can, of course, also contribute to variation in virulence. So, for example, it's common that different strains of a parasite reach different infection intensities when they infect a given host type. And this is often referred to as variation in exploitation in the evolution of virulence literature. What is less well appreciated is perhaps that similar to variation in tolerance in the host, there may also be variation among parasite types, for example, different strains of a pathogen, in how much damage they cause at a given infection intensity. And there is no established, as far as I know, there is no established term for this source of variation in virulence. So I would like to propose the term benevolence for this kind of variation. And I think it's important to distinguish these different sources of variation in virulence for several different reasons. For example, if you want to study selection on and evolution of virulence, it's important to know if the genetic variation in virulence is in the host or in the parasite. And for this reason, it's also important to have different terms for these different sources of variation. So for now, or for the rest of this talk, I will be concerned with host defense. So we'll focus on resistance and tolerance. So why then is it interesting to distinguish between these two types of defense? The key difference between these two types of defense is that resistance is, it should have a direct negative effect on the parasite, whereas tolerance does not. So this has at least two interesting consequences. First of all, resistance and tolerance have different epidemiological effects. So if a host population increases its resistance, this will reduce the prevalence of the parasite. If the tolerance increases, this should also increase the prevalence of the parasite. And resistance and tolerance also have different evolutionary consequences. So if hosts evolve resistance, this can lead to antagonistic coevolution between host and parasites, whereas if hosts evolve tolerance, this should not lead to this kind of antagonistic coevolution, at least not open-ended dynamics like if you have evolution of resistance. So since the ecological and evolutionary consequences of resistance are likely to differ, it would also be interesting to investigate what sort of the relative importance of these two types of variation among host individuals, among host populations, or among host species, and also what causes variation in resistance and tolerance. And when it comes to resistance, it's since long known that both genes and environmental factors are important. So in the case of environmental factors, it's, for example, well known that nutritional status of the host is an important determinant of its resistance to various pathogens. When it comes to tolerance, less is known about what causes or the extent of variation in tolerance and what causes it. But there are at least a few studies indicating that there is often genetic variation in tolerance. So, for example, a few years ago, when I was in Edinburgh with Andrew Reid, we did an experiment where, with rodent malaria, where different mouse strains were infected with Plasmodium chabaudi, and found that the anemia, or red blood cell density, as a function of parasite infection intensity, that this relationship, or the slope of this relationship, differed between mouse strains, indicating that there was genetic variation for tolerance to this parasite. When it comes to environmental effects on tolerance, however, there is still very little known about how nutritional status, for example, affects tolerance of hosts. So to test that, we have performed an experiment with a gut nematode, Heligmosomides, in lab mice. So in this experiment, the mice were fed either of two different diets, either normal food or low-protein food. And we also had two different mouse strains, bulb C and CBA mice, and these were selected because to represent one strain with relatively high resistance to this parasite, that is bulb C mice, and one strain that was known to have relatively low resistance. The mice were infected with two different doses of parasites or left uninfected. And then after 4 weeks, we assessed the pathology of this infection in terms of damage to the intestine. And to do that, we fed the mice with a cocktail of marker molecules of two different sizes, FITS, dextran 4, and BSA. And then we measured uptake of these molecules into the circulation in the plasma. And a normal healthy intestine is not permeable to molecules that are this large. So uptake of these molecules indicate that the intestine is damaged in some way, that is permeable to macromolecules. And a leaky intestine is bad in several ways. First of all, it will impair the ability to absorb nutrients because it's difficult to maintain the concentration gradients across the epithelium that are necessary for nutrient absorption. And a leaky intestine can also lead to uncontrolled uptake of food and microbial antigens, which in turn can lead to various inflammatory diseases. So we first tested how diet affected resistance to this parasite. So this slide shows the worm load after 4 weeks in mice with on normal food and on low protein food, and for the two different mouse strains, and also in mice fed low or high parasite doses. And as you can see, in mice fed normal food, there was the usual pattern. So bulb C mice were more resistant than CBA mice in both mice fed a high dose and mice fed a low dose. But in mice on low protein food, this difference between the strains disappeared. So in mice in poor nutritional conditions, there was no longer any difference between the strains. So this genetic difference disappeared in the bad environment. Then we tested how diet affected tolerance to this infection. So these figures show the permeability of the intestine measured as either uptake of FITS dextran 4 or BSA in the different treatment groups. And if you look at the uptake of FITS dextran 4 first, you can see that in mice fed a normal food, the dashed black and red lines, there is no effect of worm load on the permeability of the intestine. Okay? But in mice fed low protein food, the solid black and the solid red line, there is an effect of worm load, but only in one of the strains, namely bulb C. And the pattern is sort of similar in BSA, but the problem here is that there was no measurable uptake of BSA in the mice on normal food. So this means that there is an effect of nutritional status on tolerance, but only in one of the mouse strains, that is bulb C. So this shows that tolerance to this helminth infection is determined by genotype-environment interaction. And our results also indicate that the genetic differences, that is the differences between the strains in resistance or tolerance, are expressed under different environmental conditions. So in the good environment, there was a difference between the strains in resistance, but in the bad environment, on the low protein food, there was instead a genetic difference or difference between the strains in tolerance. So what we want to do next is to, first of all, investigate the mechanistic basis of tolerance, of the variation in tolerance to helminths in this system. So what's the physiological process that makes CBA mice more tolerant than bulb C mice? And I hope that a more understanding of the mechanistic basis of tolerance will improve our understanding of how this defense actually evolves. And we would also like to look in more depth at this apparently environment-dependent expression of genetic variation in resistance and tolerance. So our experiment indicated that genetic variation, now it's only 2 strains, so maybe we shouldn't talk about variation, but genetic differences in resistance are more pronounced in good environments, while genetic differences in tolerance are more pronounced in under bad environmental conditions. And with that, I am finished. I just want to thank all the others involved in this, in particular Dagmar and Olena, who did most of this work. Thank you.
Okay, so I should probably precede this by saying it's a question asked by somebody who tends to work in the field against somebody working with lab strains. But my question really is that you want to understand the genetic basis of the difference between your 2 strains, but those 2 strains are strains that exist in the lab and have not arisen. Any differences between them in tolerance have not arisen because of any selection, I don't think, of any selection for tolerance. So supposing you do what you want to do and discover the genetic difference between your strains that accounts for the difference in tolerance, my kind of cheap shot is, how much is that going to tell us about differences that might arise in a natural situation that have been driven by a need to be tolerant or not?
I agree. I agree that I think it's right to question how informative these physiological studies and things like that with inbred mice strains actually are. But I think at least that they can give a first indication about what mechanisms are important. But of course, it needs to be confirmed by studying outbred populations later. But it's easier to do these kind of experiments with lab mice. So I think that's a good first starting point, at least.
Okay, I think we need to move on. Yeah,
okay.
Very well done. And up next, we've got Joe Jackson telling us how eco-immunology.
Okay, so eco-immunology is important because the immune system is such a central determinant of individual fitness and health. And it's been understudied in terms of its interactions with the environment. And more ecologically, variation in the immune system likely determines heterogeneities in pathogen transmission.
So most of the interesting questions in this area, I suppose, interact with an understanding of what determines vertebrate immunity and immunocompetence in the wild, of course, if you're thinking about vertebrates. We know from sort of many studies in the past that there are lots of things that can affect immunity and that might affect immunity. But I think it's fair to say that we don't have a proper sort of quantitative understanding of, if you take an animal from an actual population, how its genetics, how the environment sort of add up to determine how its immune system is, or what its immune phenotype is. In sort of achieving that understanding, one big problem is not least the fact that in typical field studies, you have a very large error term, okay? So you might explain a small proportion of the variation. There's a large error term, and that's a bit of a black box. You don't know how much is due to technical or measurement error, or due to unmeasured variables. So you don't know what you're missing, really, I guess. And in addition to that, you also have the problem that explanatory variables may be confounded or collinear, and you may not sort of properly be able to detect their effects. So in this regard, it may be useful to consider temporal variation in general, and in particular seasonal variation. So every eco-immunological study that I've been involved with over the last 12 years or so has found that the passing of time, and therefore presumably change in the environment, is the major explanatory variable. It's the only really big one that explains variation in the immune phenotype. So it should be possible to take this sort of chunk of identifiable variation and partition it from the error and from other sources of variation, and to characterize it using an approach that you might term systems biology or reverse engineering. And by that I mean sort of monitoring time series in the field and the lab, and carrying out experimental manipulations of the major variables that change with season. So what I'm going to do today is tell you about a study that I've carried out with colleagues that sort of takes this approach, basically trying to sort of explain this sort of chunk of variation in a natural population. So it's based on a study originally applied for the funding 2010, I think, and it took a while to come through, and then the practical phase we've just finished recently, and what I'm going to do today is just to introduce that study and give a few preliminary results. So the model that we were using in this study was the three-spine stickleback. So it's a great natural model. It's very well studied. We know a lot about its ecology and its parasitology. It's readily accessible in the field, easily sampled, occurs in highly seasonal environments. Importantly, wild fish sort of very readily acclimate to the laboratory or to mesocosms, and it's got a lot of genetic resources, a full annotated genome, and so on. So what we did in this study was, in an initial year, we sort of did transcriptomic measurements using RNA-seq and compared two different populations, ecologically divergent sites, in winter and in summer. And in our RNA-seq data, we looked for genes that were differentially expressed across season. And we also did network analyses of co-expression to spot genes that might have a sort of high regulatory influence on seasonal changes. And from these analyses, we selected what you might call reporter genes, either with high differential expression or high influence in networks, or both, to measure using a sort of cheaper, higher throughput technology, which was real-time PCR or qPCR. So following on that first year, we sort of continued at the same sites for 2 more years, doing monthly sampling, measuring gene expression profiles for individual animals, and taking a range of other measurements, so host measurements, parasitology. We had data loggers in the field giving us 5-minutely temperature readings, and we had 10-minutely flow data for the river site. So we compared 2 different sites, a lake and a river, which are ecologically very different. And in parallel to that, we ran a sort of replicated system of 12 mesocosms that we sampled synchronously with the field sites. So these are outdoors mesocosms exposed to sort of natural temperature and photoperiod. We measured essentially the same things as in the field, but in this case with colleagues, it was also possible to do controlled infectious challenges with more than one infection system. So this is Joe Cable's group in Cardiff. And additional to this, and again in part with Joe's group in Cardiff, we were able to do a suite of laboratory experiments in which we sort of manipulated what are likely to be very important seasonal variables in the lab. So temperature, food, in which we also monitored 16S microbiota, infection challenge again, controlled infection challenges, photoperiod, and flow regimen. So all of this taken together gives us a lot of orthogonal variation that we can analyse to tease apart the different contributions of these variables to immune function in the field. And the aim is really to sort of using various types of statistical model to dissect out the effect of each of these things from our experimental manipulations, and then use the estimates so derived to parameterize a state space type model in which we link environmental variation to molecular pathways within the host, and in turn to sort of phenotypes like immunocompetence or other traits. So that's the intention. And the overall sample size for all of this is around 4,000 fish, and of these we've got gene expression profiles for around 1,300 with very few missing values amongst those. So there should be a lot of variation and a lot of power to tease things apart. Just some preliminary results, but one point worth making is that the passing of time, presumably changing the environment therefore, is the major explanatory variable in our data. This shows an ordination of 3,648 immune-associated genes in the stickleback from our RNA-seq work. There's 2 different localities, very different localities, a lake and a stream, that have been sampled in winter and summer. And the winter-summer differences are much bigger than the site differences. So the 2 winter samples there intersect each other. The summer samples just sort of shave past. There's a lot of daylight between. There's a lot of daylight between winter and summer, basically. So winter and summer is much greater than the site differences. And remember that the site differences include, all of the differences in the environment between the 2 sites, and the fact that sticklebacks between lakes and streams are typically very genetically divergent as well. So there's a lot of genetics, a lot of environment in there, but it's still outweighed by seasonal change. So really, in this system, changing the environment over time is the big determinant of how the immune system is. And in passing, I'll just say that tallies with what's been found in 2 other eco-immunological studies that I've been involved with in wild rodents that have followed time series of animals, in wood mice and in bank voles. In both cases, the passing of time greatly outweighs sort of intrinsic host variables in importance in terms of explanatory power. And I haven't shown anything for age and sex here for the sticklebacks, but again, it's their minor contributions compared to season. Also in our sort of RNA-seq data, there were some sort of mechanistic insights in these concerning, how the immune system might change across the year. Adaptive immunity genes were upregulated in the summer, and in contrast, innate immunity genes tended to undergo a complex modulation, but were predominantly upregulated in the winter. So broadly, innate immunity was upregulated in the winter, and adaptive immunity was very much upregulated in the summer. And we also found a set of genes that are associated with the suppression of adaptive responses that were also upregulated in the winter. So this really looks like a sort of strategic downward regulation of adaptive immunity in the winter. Moreover, when we constructed networks from the most seasonal genes in the sticklebacks that also had core immunological functions, very sort of highly recognisable immune system genes, when we constructed these networks, we found they had very interesting structures. So the winter bias, this is just an example network, so it's a co-expression network. The winter bias genes, shown in blue here, clustered separately from the summer bias genes, shown in yellow. And the two sort of clusters joined via a relatively small set of genes that were inferred to have high influence in the network. So the influence in the network, or the centrality, is shown by the size of these nodes. So these sort of central genes that might have a high regulatory influence on other genes, because basically this is saying that they're co-expressed with many other genes, and therefore that might have a high regulatory influence. These sort of genes bound in these two clusters remarkably contained mostly genes involved with the immunological synapse between the antigen-presenting cell and the T cell that happens at T cell activation. So without going into the fine details, that gives us a sort of potential mechanism by which adaptive responses might be limited in terms of, at the point of T cell activation and proliferation in ecological contexts. And all of these genes are quite conserved things, so beyond sort of seasonality and sticklebacks, this might be, a control point that's important in other vertebrates. So conclusions, as I say, mostly what I've spoken about is preliminary analyses, and it's a sort of a watch this space sort of situation. But most immunological variation, I think we can say quite definitely, is driven by the environment. Okay. I would say that decomposing seasonal or temporal variation, where you've got an identifiable chunk of variation that you can aim at, is a more tractable way to understand the controllers of immunity in the field. And also, it appears that there may be mechanisms limiting T cell activation that provide a control point for limiting adaptive immunity in sticklebacks. But potentially that might be a more conserved mechanism relevant in other systems. And finally, we were asked to sort of make thought-provoking comments. I'm not sure if this is or not, but I'd say that combining field monitoring, lab experiments, mesocosm experiments, genome-wide expression measurements and network analyses, and also direct measurement of disease progression in challenge infections, and then analysing them all with sort of system biology type modelling is a powerful approach that will allow conclusive studies. So it will allow you to sort of tease apart sort of variation that otherwise might be sort of confounded and not visible in field studies. Okay, thanks.
Great, thank you very much. I think we're going to have to move on. I'm sorry. Can we take the next talk and then it's time to maybe come back and try I'll try and stick to the schedule as much as possible. So we'll move straight on to Mark Heine, who will tell us all about the determinants of immune function in one house.
Well, thank you very much. Thank you for the opportunity to talk here. I'd like to present the work that we've been doing for the last few years to understand the immune function of wild mice. So I'm using a Mac, which I think has caused the problem already. Okay.
Just wondering for your summer winter, is that 6 months versus 6 months?
So it's comparing September and February, which actually turn out to be the sort of the seasonal extremes. Yeah, but it's not sort of the peak of summer in the sense of photo period, but rather towards the end of the summer, which is more environmental.
Okay. All right. Okay. So thank you very much, as I said, for the opportunity to speak. I should say before I go any further, I'm representing a whole group of people who have done all of this work. This group of people, well, excluding myself, in Bristol, and our immunologist colleagues in London. It's very much a joint project. So the questions we set out to ask were actually what immune responses do wild animals make? And then what drives and what constrains the responses that they do make. And actually, just in case we do have time, I'll perhaps give you the answer before we start, and then I'll present the data I hope to persuade you of these answers. So what we find actually is these mice have a highly activated immune response, presumably due to very high antigenic exposure, but they have very significantly depressed cytokine responses. We also find that these different populations of mice, even at a very small scale, differ immunologically, but this is not due to the genetics of the mice. And we find that particularly mouse age, it both drives and it constrains the immune response that these mice make. So first of all, let's just tell you what we did. We studied the wild version of the laboratory mouse, Mus musculus domesticus, because essentially all that we know about vertebrate immunology, or 90% of it, I guess, comes from study of this species. And so we went to chase the wild version of these, and we particularly chose this species because we could use all the immunological tools that are available for the wild species, from lab mice with these wild individuals. So it would give us an ability to look at the immune system in really quite considerable detail. As Paul has already said, the underlying context for this work is that immune responses are costly, and I won't repeat the evidence for that. And so we're thinking about, well, what are the costs that these animals actually have in the wild, and what are the effects, therefore, on their immune responses? What we did over a period of 2 years was we trapped mice in and around Bristol. These are mice that live commensally, so they live in farm outbuildings typically, or other agricultural settings. As we sampled around Bristol on quite a small scale, somewhere in Gloucestershire as well, and we had 2 outstations, as it were, this island of Skocum in far west Wales, and also the London Underground. We caught 500 mice over a couple of years. We killed those mice so we could do their immunology properly. And we've measured their concentrations of the various immunoglobulins. We've taken their spleens and measured their, using, for instance, activated cell sorting, the types of cells that they have in the spleens, and a lot of information about the activation status and the status of those cells. We've also then in vitro stimulated those splenocytes with some molecules to simulate microbial infection, and then looked at the cytokine responses. And of course, we've measured other things about these mice as well, some aspects of their morphometrics, their body condition, their infections, and their genetics. And have tried to explain these immune responses from these measures. So first of all, just comparing these laboratory mice, the wild mice with these laboratory mice, they do differ, as one would expect. So wild mice are a little bit smaller. They're about 1.5 times, lab mice about 1.5 times bigger or heavier than the wild mice. These wild animals live a very short life. The median lifespan is 7 weeks, and 75% of animals are 12 weeks or younger. But interestingly, they have disproportionately small spleens. So this lab mouse here is 1.5 times bigger than a wild mouse, but it's got 5 times the number of spleen cells. So I wonder whether actually laboratory mice have been inadvertently selected by immunologists to have a large and highly tractable spleens. We've got a lot of immune characterization of these mice, but I'll just give you 3 bits of data just to make the general point. to answer the question, what immune responses do these animals make? So first of all, if we look at the total immunoglobulin G that these animals have in their serum, comparing males and females, where the green here is, will be the wild mice and the non-green, the white, will be laboratory mice. There's an enormously high concentration of circulating antibodies in these laboratory mice. This is on a log scale. And presumably this is because these animals are exposed to a very high antigenic load through their remarkably short life, but they have an awful lot of antibody in their serum, and also immunoglobulin A in their feces as well. We see the same pattern actually with respect to the aspects of the cellular compartment of these animals. So here's just one example. So this is for T helper cells, and it's asking what the flavor of those T helper cells is, whether they're in the naive state, so they're not antigen exposed, whether they have a memory function or an effector function. And what one finds is the wild animals have cells, T helper cells, that are affected because they've been exposed to antigen, so they're responding. And we see this through CD8 cells and lots of other cells as well, that these cells are active, they're doing stuff because presumably they're exposed to a lot of antigen. In contrast, the cytokines we find are depressed compared with those in wild mice. So here we've taken the spleen cells of these animals, and this is the control here, stimulated them with these various molecules, and in this case measured this cytokine that they produce. And in many cases there aren't differences between laboratory and wild mice for 9 cytokines across these 4 molecules here. But where there are the differences, the wild mice usually have lower responses, as you can see here. So what we see is these animals have highly activated immune systems, but their cytokine responses are depressed. And I think the cytokine responses are depressed because actually if they had cytokine responses that were commensurate with the elevated cellular stimulation, that would be immunopathological to the mice. So there has to be downregulation of these cytokine responses to preserve the health and the fitness of these mice. So next we wanted to begin to think about, well, these are the immune responses these animals are making. What's controlling this? What's driving? What are constraining these responses? So we start, just very briefly as an aside, in natural killer cells in these mice, it was known from studies of lab mice, they have this molecule on the surface, LY49H, which is the receptor for cells that when they're infected with cytomegalovirus, this is the molecule produced by the virus. And this molecule is necessary for mice to be resistant to this infection. And if you give this infection to mice that have a deletion in this gene, then these mice are completely susceptible to this infection. Interestingly, we find about 20% of our mice are homozygous for a deletion in the gene that produces that molecule, suggesting that the virus infection is probably very different in the wild. And what the laboratory mice tells us is a lethal infection would appear not to be true in a wild setting. Okay, so what drives and what constrains these immune responses? Because we had these different sites, we were first of all interested in looking at the genetic difference between these mice. So we genotyped them at about 1,000 loci, and then from this constructed a tree of the genetic relationship between these mice using these autosomal loci, where the sites, these are each branch is a tree, is a mouse, each branch is a tree, each branch is a mouse, and the color coding is the site the mice are from. This compared with the laboratory mice here, so they're clearly mice. Would anyone like to make a hazard guess where the mice from the London Underground are? Okay, they're down here. Okay, so we see the mice from Skocum, the far west site, are quite different from the rest of mice, and actually slightly more inbred because these branches are shorter. The dominant pattern, though, is actually mice clustered by the site they come from. So mice are really quite different at each site. London is about 100 miles away from Bristol, so you might expect these mice to be different, and indeed they are. They have an FST value of about 0.4. Perhaps the more surprising thing, all of these sites, many of which are close together in and around Bristol, there's as much difference between these very local sites as well. So there's very great heterogeneity between closely located sites. Because of this, we're interested in asking whether what the immune flavour, if you like, was of these mice. And the way we approached this was following a study in humans where they calculated the immune distance, which used a principal component analysis of a number of immune measures, and then from that calculated the distance between individual mice, both in our case within and between sites. So here we've got a plot of the immune distance between the mice, where each dot is one of our sample sites. The size of the dot is how heterogeneous or homogeneous the mice are immunologically, and the distance on these axes is the difference between the different sites. And what this shows here is that many sites are immunologically quite similar, but some sites are really very different. But the degree of immune heterogeneity varies very substantially by sites as well. This immune distance has nothing to do with geographical distance, and it also has nothing to do with genetic distance. So there's substantial immunological differences between sites for some other reason. And in view of this, this heterogeneity, to ask the question about what's driving and what's constraining these immune responses, we next just focused on one site, which is this site in here. And we use structural equation modeling to ask what drives immunity. This should actually say immune function, but it doesn't fit in so easily, so I use immunity. And from the literature, and again, as Paul said in his introductory talk, we thought about things like that were likely to affect the immune function, being the condition of animals measured as the scaled mass index, the season when we caught them, the infections, which actually are a collection of microbial-only infections they have. These are parasitologically very uninteresting mice, or they're interesting because they have so few parasites, and the age of the mice. And so we therefore have this full causal diagram for these mice. So we hype these all the possible interactions between these factors we measured in square boxes here, and this so-called latent variable, this thing we were trying to explain the immune response of these animals. It took us an awful long time to work through these data, and one part of the resolution was actually trying to find the right measures of immune responses and actually dealing with different, the conclusion was to, or the resolution was to deal with different compartments of the immune response. And this is what we find for mice at our principal study site here. So it's the same diagram as you've just seen, but it's done separately for males and females. And so this is here is asking what is driving the adaptive cellular immune response, measures T helper cells, T cytotoxic cells, and the B cells here. So you see the principal effectors, and the reds are negative covariants, and the thickness of the line is showing the strength of those, and blue are positive covariance effects. Again, the thickness of the line is showing the effect of those. So we have two major factors going on here. First of all, age suppresses these immune responses. So this is presumably some sort of immunosenescence, even in these very short-lived animals. But body condition actually promotes the immune response. And therefore, in females, the age has a divergent effect because age reduces these immune responses by these measures, but it improves condition, and condition improves the immune response there. We have some effects of season, as Joe was just alluding to in his talk. Infection actually is a rather small effect and actually apparently absent in males. We see a very similar pattern when we look at the innate immune response here. Now our measures are natural killer cells, macrophages, neutrophils, and dendritic cells. So the same pattern, but there's a quantitative change in these various effects, and we lose the seasonal effect here. But still, we have the dominant effects of age reducing these immune measures, condition promoting it, and therefore age tying these two things together in females. We have a very different pattern in when we consider the antibody responses here, so the serum IgE and A and the fecal IgA. And here, the dominant effect is of age. So as animals age, they acquire more of these antibody responses, and that's because, antibody responses are cumulative. But again, it's rather a small effect infection, and we still have this difference between males and females. So therefore, I hope I now persuaded you of the truthfulness of these answers, that these animals have a highly activated immune system, but a depressed cytokine responses. And presumably that depression of the cytokine responses is the animal's ways to protect themselves from potential immunopathology. We find that these populations differ immunologically, so that's the immune distance, but not because of their genetics or indeed their physical location to each other. But I therefore suspect it's because of very local ecological effects. And what we find is that mice age both drives and constrains immune responses via the conditions or via body condition. But this differs across different immune compartments. And therefore, I'll finish by thanking our funders and all these various people who have contributed to this work. And thank you for your attention.
Maybe time for one quick question. Go on.
I think it might be something to do with their infections. we haven't got a good handle on that, I'd say, but they seem to have slightly different patterns of infection that might be driving it.
And you didn't find any worm infection?
Sorry, they all have pinworm, except Skocum and London, but it's universal elsewhere. But there are very few other nematodes. They have mites, but those are lice, but they have no effect at all. So that's just 7 microbial infections we have in there.
Thanks very much, Mark. So we should move on now to Richard Francis, staying I think with the mouse thing. Tell us about whipworms.
Okay, first of all, I'd like to say thank you very much for inviting me. I make no apologies, I'm an experimental immunologist, so I am interested in mechanisms, but what I'm going to talk to you about today is the work we've been doing with this particular parasite. I want to give you a flavour for what you can do in the laboratory, what we found out, and I'd welcome any comments or suggestions about how we take this forward into the broader world. So we work with parasites of the genus Trichuris, which is known as whipworm, and here you can see it's an intestinal roundworm, and it looks like a whip, which is why it's called whipworm. And Trichuris is a very successful genus. It has all these. I spent one afternoon looking in the literature at finding animals that had Trichuris documented. All these types of organisms have Trichuris. It's radiated out, and it's found in man, which is a major infection in man, but also in the mouse. And this is what we work with down here in the mouse for obvious reasons. Because it's a human infection, people have looked into the past, and it's, in essence, like you would expect, it's been with us for a long time. So you'll have all heard of Ötzi the Iceman. Well, he had Trichuris, but also close to home, just down the road from where I live, the Lindau man also had Trichuris. Wherever you look, I can go anywhere in the world and I can look in the paleobiology, I can find people that have had Trichuris. So it's been very successful. It's a really good parasite. Where does it live? It lives in the large intestine, this nematode parasite, and its preferred site of infection is the cecum, whatever it looks like. And this is the first known drawing of a human cecum by Leonardo da Vinci. And of course, as you might expect, Trichuris was prevalent in Renaissance Italy, so maybe Leonardo had Trichuris as well. To orientate you as to what the parasite looked like and the infection, this is what a mouse or a human would ingest if it was a human egg. They all look the same, this sort of rugby ball with these 2 plugs at the end, and you can see the L1 larvae inside. These hatch, they don't look like whipworms when they're in the L1 stage, but over a period of a month in the mouse and a little bit longer in the pig or the human, they develop into parasites which are about 2 centimeters in length as an adult, a little bit bigger in the other species, but not that much bigger. And you can see that their niche is embedded in the epithelium, their thin whip-like end in the epithelium of the gut. So this is a really tight association, all right? You can't see the mucus here, that's come off, but it's sewn into the intestine. And animals and people will have these kinds of infections for most of their lives. So it's a definite invasion, so you might think in terms of cost what effect it might have. So this is just a nice older picture. You can see it's snaking its way, its epithelial tunnel, and the back ends of this dioecious species stick out into the lumen of the gut. They mate, and the females lay eggs that pass out with the feces. You have an embryonation period, and then they become infected. This just gives you an idea, looking at a cross-section of the cecum here, in this thin whip-like end is embedded. These are transverse sections through the worm, and the fat ends hanging out into the gut here with the eggs inside. So it really is a tight association with the host tissue. Now, we've spent a long time trying to understand the immune response to this kind of parasite, and we've been doing it in inbred mice, as you might expect, and us and a lot of other laboratories have quite a detailed understanding of the different cell populations and mechanisms of resistance and susceptibility. Most inbred strains of mice will expel a large bolus infection of Trichuris, and when they do, they make this kind of T cell response in these T helper cells of a particular subset. The Th2 set are really important. But basically, if a mouse makes a lot of this cytokine, interleukin-13, that comes from these cells, the animal will be resistant. But there are some inbred strains which don't make this kind of response, and actually harbour a chronic infection of lots of worms. They make no less robust an immune response, but it's really quite different, and it's dominated by a different subset of T helper cells, ones that make gamma interferon. And gamma interferon is more associated with, if you like, antiviral or antibacterial infections. So there's this underlying difference in mouse strains as to why you're resistant or susceptible. What's also interesting in Trichuris is actually you can change the resistance status and the immunological profile that you get based on the dose of infection that you give. So most strains, as I've mentioned, and we've looked at lots of different inbred strains, if you give them a moderate to high dose, they'll throw the parasites out, make a Th2 response. But actually, if you give those same strains, instead of 200 eggs, you give them 20 eggs, they'll become susceptible and they'll switch on a Th1 response. But of course, in trying to understand acquired immunity in a slightly more realistic approach, if we consider the idea that infection by a single high dose is likely to be a rare event in nature, as is a single low dose infection, we've started to look at multiple low doses. This is still quite rarefied, obviously, from the field, but actually, multiple repeated low doses, what happens? What happens in this trickle infection? And so we set up this system where we've now established quite a nice 9-dose infection over an 11-week period, and we've done an enormous immunological phenotype of these animals. And actually what you see is that after this trickle infection, to begin with, you start to build up infection, and this is looking at the different life cycle stages of the parasite, but eventually immunity kicks in, and by week 11, you get partial immunity. And this is sort of the thing that you see in outbred human populations infected with soil-transmitted helminths, including Trichuris. You never get complete immunity after repeated exposure. You get partial immunity, but you do get some immunity. So what is the basis of this immunity? Well, I'm just highlighting the things here that we know are associated with resistance. And in essence, without going into great detail, when immunity kicks in between weeks 9 and 11, you get exactly the sort of immune effector mechanisms and cytokine responses being elevated that we saw after a single high-dose infection. So the mechanisms of immunity seem to be similar even after trickle infections. And in fact, we know that the CD4 cell is important because you can immunologically go in at week 9 and inject an antibody to remove the CD4 cells. And when you do that, when immunity kicks in, then all those changes disappear. So I think CD4-mediated immunity, even after generating immunity for trickle infections, is quite important. But we're also interested in the host-parasite relationship in a little bit more detail, and to remind you that we are considering a parasite that lives in the cecum, which is full of microbes. The parasite inhabits here, and we were very interested in why the parasite, when the egg goes in here, why the parasites live here. And essentially, we observed that actually the majority of the hatching of the parasite takes place in the terminal ileum, and this is about after 90 minutes of infection in the host. And this occurs, the majority of it, as soon as the eggs hit the microflora in the gut. And in fact, you can recapitulate this in vitro by taking the cecal contents and looking at hatching overnight. And even better, you can just take bacteria out the lab and cause the eggs to hatch. It's not the filtrator, not the broth they're grown in. And you can get really efficient hatching with bacteria, a variety of species of bacteria. And because they're genetically tractable, you can label the bacteria, and you can see very nicely that they attach to the polar plugs where the parasite crawls out. E. coli uses fimbriae to attach, and mannose is important in that binding, and if you add mannose, you inhibit hatching. And in fact, you can actually genetically knock out the fimbriae in the bacteria, you get no hatching. You knock the fimbriae back in and you get the hatching, and that's shown here. If you knock out the fimbriae and add it to eggs, you get no binding to the egg and you get actually no hatching. Here, you can actually see that the bacteria, when you put the fimbriae back in, start to stick to each other on the edge. And in this situation, you do get hatching. So I think the bacteria, the environment, dictates where the parasite hatches. And we know this occurs in vivo as well, because if you treat animals with antibiotics, you reduce the hatching either in immunocompetent strains or in animals with no immune system. So if the host affects the parasite and dictates where it hatches, does the parasite affect the host flora? We did this experiment where we monitored flora in the infection and after a period of time cleared the parasites out with a drug to see what effect the parasite had. And yes, it does. And in essence, because for the sake of time, what you see during infection is you get a loss of diversity of bacterial species during chronic low-dose infection. When you give a drug, then it slowly starts to return, the diversity slowly starts to return towards the naive that you saw to begin with. So what are the consequences of this changing flora? Does that have any effect? And we set up an experiment where we gave a chronic infection and superimposed another low-dose infection, and we wanted to see whether the dysbiotic gut that you saw affected whether the parasites would hatch and establish in the second infection, and in fact, it does have an effect. So here you see the number of the adult worms from a primary infection and the number of worms in a single infection animal that get in, looked at L2 and L4. Here you can see adults plus L2s in the dysbiotic gut, or looking at the L4 stage. The eggs don't hatch as well in the dysbiotic gut. If you take the dysbiotic gut contents and do in vitro hatching, the eggs don't hatch as good. And if you remove the parasites with a drug and use only the microflora without the worms, again, you don't get as good hatching and establishment of parasites. And you see the same in the absence of the adaptive immune response. So the parasite uses the bugs to get in and hatch. It changes the biota of the host, and that impacts on whether the parasite can establish the subsequent infection. So maybe it's a strategy evolved by the parasite to prevent large numbers establishing. But of course, finally, we shouldn't forget that the parasite itself, as a nematode, has a gastrointestinal tract, and you can actually look at the microbiota in the gastrointestinal tract of the nematode and compare it with the infected mice. And in essence, you see a big difference. You don't need to look in detail at the things at the bottom, but that last line here is the biota from the worm's gut as opposed to the naive mouse or the infected mouse. And so we see quite a lot of differences, particularly in the proteobacterial proportions. So the parasite seems to pick up a flora from the host and adapt it to its own needs. And in fact, if you take parasites out and you incubate them, adult parasites, in a cocktail of antibiotics, they die much quicker than they do in media alone. But if you take the L1 larvae out of the eggs and they don't have a microbiota, they're sterile when they go in, antibiotics don't have any differential effect, so it's not a direct antiparasitic effect. And infection of these kinds of parasites in animals that have no biota is far from productive. So to summarize, we think there's a big interaction between the immune system, the microflora, and the dosing, which determines the overall survival and establishment of this kind of nematode. Now, how this applies to the wild is another thing to think about, but it does extend a little bit simply immunity and laboratory studies and infection. As always, I don't do very little other than infect mice, but there's a large team in my lab that helps. The biota stuff of the worm was done by Emily White, which is very much a collaborative project with Ian Roberts and Ashley Holden, who are microbiologists in Manchester. And I'd like to thank our animal unit and our funders for allowing the work. Thank you. So, yeah, it's a very good question. Nobody knows. We don't know. So, there's so many things that impinge on whether you generate a strong Th2 response quickly. And as yet, we've looked at numerous cytokines, numerous different cell populations, following low dose and high dose to try and get a clue. And we don't have a definitive answer. But then again, for most immune responses in any term, from immunity to autoimmunity to transplantation to cancer biology, we don't understand why some individuals make a Th1 dominated response or a Th2. There's very little information, hard information, to tell us what the critical thing is. It may not be one critical thing, it may be context and time dependent. So I can't answer that, I'm afraid.
I think we should, sorry, I think we do need to move on. So plenty of time for questions and answers. Thanks, Richard. So next, we've got Simon Bamian, who's
Could you have the first slide, please? Oh, okay. Well, you're going to see all the talk, right? So I think from this morning's session, it's quite obvious that it would be highly desirable to be able to combine the power of lab systems with the relevance of natural populations. And studying immunity in natural populations does, of course, come with its hurdles. Typically, unless you're working on house mice, that means that you actually only have a very narrow set of immunological tools to work with. Obviously, you cannot have genetic knockouts, but even the monoclonal antibody situation usually isn't all that good for most species other than mice. But by far the biggest problem when studying natural populations is the substantial heterogeneity and variability. And the normal way of going around that problem is increasing your sample sizes. But that isn't always easy, especially if you work on some protected species, for instance. And by and large, and for the foreseeable future, our knowledge of species of interest is actually very limited. Very few published genomes are available. For example, I don't think there is a published genome for badgers, surprisingly. And of course, we have very little information about the internal physiological state and immunological history of the individuals. However, and as Joe has alluded to earlier, there is much hope that high-throughput omics technologies might be able to level the playing field and allow us to start to interrogate more deeply species that were so far difficult to study. But of course, what we would like is to build an immunological approach that is both tractable and generalizable. Tractable in that it would provide detailed molecular and cellular information when needed, and that is relevant to fitness of the hosts, and we would like that information to be both specific and repeatable. And generalizable in the sense that the methods and the inferences we make about those individuals under study should be generalizable to other populations, possibly other species as well. So I mentioned omics and transcriptomics in particular are possibly promising, but they do come along with their own set of problems and hurdles. One of the biggest ones being the curse of dimensionality, in that when you sequence a transcriptome, you get thousands and thousands of variables essentially, and you typically have very low numbers of samples. And those variables tend to be quite autocorrelated and very weak predictors of the phenotypic outcome of interest. Furthermore, transcriptomics in particular suffer from the problem of looking at RNA only, so it's very hard for some genes to infer how RNA expression relates to protein availability and activation. Essentially, we need to account for the difference in RNA expression regulation between different genes for a given individual. However, I think using RNA has specific advantages over proteomics or metabolomics in that at least we have a sequence of a gene that is more highly tractable than the other methods. And then immunology itself comes with its own set of problems, and we've heard about this morning. The immune system and immune genes tend to be highly redundant, or there's a lot of redundancy built in the immune system, whereby different genes provide similar function on the surface. The expression of these immune genes is nonlinear, as mentioned, so you've got some genes that are regulated during transcription and some during translation, and we need to account for that during the analysis. We can't ignore it for all the genes. The relation between copy number and function tends to vary between the genes and between individuals. And something that's specifically important in the immune system is that sometimes it is the ratio between two different genes that will have a bigger impact on the overall function than the absolute copy number, for instance, or expression level of those genes. So we would need to find a way to preserve the ratios between, say, effectors and regulators in order to get a closer match to immune function. So the approach that we've been taking, we've tried to account or to address all those problems. What we have been working on basically is using a clustering method to cluster together all genes that seem to behave in the same way, and using a clustering method that preserves the so-called geographic difference between gene expressions, that is to preserve the relationships between genes within individuals. I mentioned the nonlinearity problem of immune gene expression, and because most genes are probably not expressed in a linear fashion with increasing doses of parasites, for instance, what we've chosen to do is base most of our analyses and our approach using non-parametric approaches which don't make any assumptions about how genes or the distribution of expression is generated. To address the problem of very high numbers of features with low sample sizes, we choose algorithms that select important features and eliminate those that are less informative about the trait we're measuring. We favor approaches that allow us to quantify the importance of specific features in our predictions and in our analysis. In other words, we shy away from methods that are black box, such as principal component analyses that tend to muddy the waters and make biological inference harder. And to ensure that our analysis of the immune profiles is as generalizable as possible, we use algorithms that penalize overfitting, so that we try to minimize the amount of noise that we're fitting, and we use various methods of cross-validation to ensure that. In other words, we've been using supervised statistical learning as a method of analyzing immune profiles. The idea basically is that we let the computer derive from observed data the appropriate function that maps immune profiles to desired outcomes such as parasite burdens. So the study system we've been using is one that you're probably very familiar with. It's the wood mouse and Helimosmoides polygyrus parasite. These data are collected in natural populations, and I'm deliberately ignoring all other factors. Right? I'm only focusing on immune profiles as measured as RNA expression and H. polygyrus burdens. So what we did for this experiment was to capture the mice and treat half of them with antihelminthics, so we're manipulating the parasite dose. And then 2 weeks later, we sample them and take various measures. And the ones that I'll be focusing on are really the RNA expression in the spleen of these individuals, fecal egg counts, and the worm burden in the guts. So they are terminally sampled at 2 weeks after treatment. And essentially what we see on the parasite side, well, first of all, in total, we sequenced transcriptomes from 35 individuals, distributed as so, roughly balanced groups between treated and controls. And the infection profiles for polygyrus in the population, show that 70% of the individuals were infected by H. polygyrus. And you see the typical negative binomial type distributions of parasite counts in these populations. So most individuals are infected with low doses, and you have some that are infected with high numbers of worms. And we also saw that the effect of treatment was significant, and there was a substantial reduction in worm burdens in the treated individuals, and a near complete elimination of eggs in the feces of the treated individuals. So, taking the RNA and sequencing the RNA, we were faced with a problem that is probably relevant to anyone setting out to do transcriptomes in these non-standard species. And what I'm going to give you is an example of transcriptomics with 3 different starting points. The first one, which is species, for instance, for which you do not have a genome at all, and where there are not that many species that have been sequenced that are closely related. So in that case, what we did was an unguided assembly. So we need to build a reference to be able to count gene expression, to quantify gene expression. So the first step is having a reference to which we should map these transcripts. Now, to build that reference, using the unguided assembly, basically you only rely on the transcriptome itself and you hope for the best. When you have a species available or a genome available from a species that is close enough, closely related, you can use that to guide the assembly of your reference transcript. So in that case, a guided assembly, and what we did here was guided the assembly of the epidermis sequences using the mus musculus, so the house mouse genome. And if you're lucky enough to have a genome that is available, then you can simply map all your transcripts to the conspecific genome. And we did this using a genome that Steve Patterson from Liverpool kindly let us use. So basically, once you've got your reference sequence, you map all your transcripts to that reference and you quantify them. You count essentially the number of copies of each gene. And if you do that with the unguided assembly, what we ended up with was 324,000 transcripts. Obviously, for an organism that's probably got about 15,000 genes, 20,000 if you count the isoforms, that's a bit of a problem. And obviously, that's 324,000 variables with only 35 individuals. So we have a massive P over N problem, 9,000 times. So the curse of dimensionality is going to hit us hard for that one. The guided assembly using the mouse genome, the house mouse genome, gives us a slightly better count of 84,000 transcripts, which is still very high, but slightly better. And we have slightly more confidence in how the assembly was made and the inferences that we might be able to make from that. And if you look at the conspecific reference genome situation, you've got 18,000 transcripts, which is about what you'd expect. And of course, given our sample sizes, it's still quite a lot more variables than samples, but it's as good as we can get. So usually the bioinformatic approach is to do the sequencing and then start plotting in an unsupervised approach your individuals and see how they cluster. In this case, to see if they clustered according to treatment. And your bioinformaticians usually come back and say, well, you've got a problem, nothing's clustering. And well, if you're hopeful, there might be some mild clustering there, but nothing much to work on. But this is an unsupervised approach, right? It's only looking at transcription profiles of the individuals without any other information. But it turns out that we do have other information, and we'll be using that. But before we get there, we have to deal with this large number of transcripts from the transcriptome, most of which are probably, or a large proportion of which are probably redundant and collinear. So what we do there is simply to cluster all the genes that seem to behave in the same way, to try and minimize the uninformative variation. So when you start clustering, you have a wide variety of options available in how you actually cluster your data. And the one that we chose is density-based agglomeration, essentially, so DB scan. these are just toy data showing the performance of different algorithms, clustering algorithms on different shapes of data. We picked this one because it allows clustering based on density, which basically allows you to agglomerate together genes that have very unusual transcription profiles and that don't necessarily need to be globular or circular. And then what we did with the statistical learning is to try and map our transcription profiles to variables of interest. In this case, treatment or sex. I'll be ignoring sex. I'll just be talking about treatment, worm burden, and worm fecundity. And G here is essentially the space of all possible functions that could map the X's to the Y's. And what we'd done there was to compare various algorithms and rank them according to their accuracy in their prediction and the ability to map the transcriptome profiles to these variables of interest. And of those, we went for the variables that preserve the individual information of each of the transcripts and are based on a non-parametric method, so we used extreme gradient boosted trees. So it's based on decision trees and partitioning of the data set with a non-parametric approach. So here are some of the results. Using the easiest set of transcriptome data first, the reads that are mapped to the epidermis genome, we get a relatively low number of clusters that predict the treatment status of the mice. Likewise, we're pretty good at predicting parasite burden, and predicting egg production is not all that great. So when you see bars of equal lengths, that means that they are all equally informative in predicting the outcome, which is actually means it's not very informative. Right? And this is just a representation of the overlap of the genes between these different systems. So what we can do, because of the choice of our algorithms, is pull out the genes from each of these individual clusters and then try and infer function from those. This is the situation using the house mice guided reference. Again, we're doing pretty well with treatment status, predicting worm burden is okay, and completely useless at predicting egg production. And using the unguided reference, which supposedly is the hardest situation with all this 324,000 variables, we're still actually doing pretty well. We've got good predictors of treatment status and of worm burden, and okay predictors of egg production. So not having a genome of your species is not the end of the world. You can actually make good use of it. And just briefly, not to go into too many mechanistic details, but you can pull out some interesting immune genes. Here, in this case, TNF14, which is part of the TNF family, is one of the genes that differs between treatments. And here you have a retinoic acid receptor, which is involved essentially in regulatory T cell development, which decreases with increasing worm burdens. And for worm egg production, here we have the TNF again, the same ones here, which is very, very weakly associated with egg parasites. What this illustrates is that each individual gene is probably pretty weak in these situations, but the advantage of the machine learning approach is to combine a lot of very weak predictors to make a much better and stronger prediction in the agglomerate. And it also allows to uncover some surprising features. For instance, here, this is a gene involved in circadian rhythms. No idea what the relation between antiemetic treatment and circadian rhythms might be, but you know. Here we have an association with a gene that's involved in oogenesis, so in females, the ability to produce ovules. And here we found another surprising one, which is a gene which seems to map specifically to yeast, which is associated with egg burdens for some reason. So to summarize very briefly, using these supervised statistical learning approaches allows mapping transcription profiles to pretty high-level phenotypic traits, so that mapping of transcriptome to phenotype seems to work fairly well. Because we're using machine learning type approaches, actually the models that we have trained on the small set of data will improve over time as we add more data to the same models, and they'll benefit from sequencing in more populations. But as we saw this morning, having some longitudinal information and longitudinal transcriptomes would, of course, be much more powerful in identifying causal relationships between gene expression and parasite outcomes. And, of course, taking a whole transcriptome approach rather than having a narrow set of target genes allows you to make some unexpected discoveries. Let's see where this goes, but yeah, that's the opening for the future. And, of course, this was done with many people across Glasgow and Edinburgh, and had good help from bioinformatics specialists, Graham Hamilton and Wayne. Thank you.
Sandy, I don't think we've got time for questions, and we're so close to lunch. I think we should just move very well. Thanks, Sandy. So we'll bring it to a resounding finish. Got Andrew Gray. Can you tell us about heterogeneity in music process?
So thank you very much to Andy and our other gracious local hosts, and to the BES for the invitation. I'm really happy to be part of this symposium. Now, is this something? Oh, I'm showing Simon's candidate genes. What do I have to do? Oh, keep clicking? Don't ask me any questions about this, please. We're back on track. Okay, I also want to thank my co-authors and collaborators on the central results I'm going to present to you today, especially past and present members at my lab, Jackie, Sarah, Tina, and Romain in particular, and also members of the lab of my collaborator, Peng Lok at NYU. So like many of my fellow speakers this morning, I'm very keen to understand causes of host heterogeneity in this wormy world, a term coined in 1947, as true in the 20-teens as it was then. Over 1 billion people and countless animals have bellies full of gastrointestinal nematodes, and I'm very keen to understand why. Like many of you in this room, I'm particularly keen to understand the extent to which costs of immune defense can help to explain that heterogeneity. Now, I, through most of my career, have studied immunopathological costs of defense, so collateral damage caused by immune systems. And so to answer Paul's question, that's why in the Soe sheep study he mentioned we were initially starting with the anti-nuclear antibodies, the autoimmune antibodies. But I'm very much also interested in understanding heterogeneity in defense against parasites, and in particular, moving toward resource costs of immune defense in recent years. So one of my favorite factoids in biology is that people have done careful work in vitro to count the rate at which highly activated plasma cells secretes antibodies, and it's become clear that they can secrete thousands of antibodies per second and sustain that for weeks. So just the amino acid, the investment, essentially, in that kind of production rate of an effector like an antibody is what just mind-blowing. And of course, ecological perspective suggests those are amino acids the host can't invest in things like growth and reproduction. But that's a very Jack Sprat lean view of resources, right? So if you prefer, you could also think about fat as being something that a host can burn to invest in immunity. And there's also increasing evidence that fat itself is an immunological tissue, almost lymphoid. And so these are just two complementary ways of saying the same thing Paul started us out with, which is we think there is going to be a big resource cost to investing in immune defense, and you might imagine that that might ramify into the maintenance of host heterogeneity. And this, of course, harkens back to the foundational paper of Eco-immunology. So my own approach to shortening the hyphen between eco and immunology, especially in recent years, has been to study these resource costs of defense in what I call mesocosms. I'll tell you briefly about one and then go into greater detail on the other. But why mesocosms? I think that they're extremely useful for slightly collapsing the huge complexity of these complex systems. Also, I choose systems where it's nematodes infecting mammals, where actually the mammals allow me to have a pretty amazing toolkit to deploy. So it's not only the laboratory mouse, but also species that have gotten a lot of veterinary immunological attention, such as sheep. So I consider the island on which the Soay sheep live, St. Kilda in the Outer Hebrides, or the island of Herta, I consider that a bit of a mesocosm in that it's a closed system to migration, that sort of thing. It's also free of predators to a large extent, and so it has that slightly reduced complexity. And we can actually observe the internal dynamics of this system over time. And Josephine Pemberton and co. Have been the visionaries who established and continue to lead this project. Last year was its 30th anniversary, this long-term project. I just wanted to show you this plot, a long-term time series of host densities, booming and busting. That's the gray data projected onto the right axis. And then the black data projected onto the left are the average gastrointestinal nematode burdens in the lambs born in each year, sort of using lambs as a sentinel for intensity of nematode transmission. It's fecal orally transmitted. So we spent a lot of time staring at this plot and trying to understand what the causes of these dynamics might be, especially focusing on the sheep-nematode interaction. We don't only stare at the plot, we actually make time to do some immunoparasitology, and we've been successful in making sense of both resistance variation, so the variation of the ability of hosts to clear nematodes during that highlighted study period there, as well as variation in their ability to tolerate nematodes over the whole study period. I wanted to highlight that especially the resistance variation work suggests that this environmental heterogeneity, if you will, sort of biotic environment, how many parasites are there being transmitted in the population in a given year? How many competitors are there for the sheep when seeking food? Those seem to be incredibly important in the maintenance of resistance variation in this population. But most closely to the point of the resource costs issue that I want to focus on today, Romain Garnier, when he was a postdoc working with me, led a couple of studies just in this final year where we had a really big population crash, quantifying protein nutritional plane in individual sheep, and made some really exciting findings. So first of all, an individual's ability to maintain protein nutritional plane in the face of all of this booming and busting does seem to promote condition and survival, but we can also detect a trade-off against resistance that's measured in antibodies. So we really are, it looks like in this system, able to quantify a protein resource versus antibody trade-off. And I love the Soay sheep, but I'm also an experimentalist at heart, so what I want to spend the rest of the time on this morning, or afternoon, it definitely feels like afternoon by body time. What I would like to focus on for the rest of the time is a new system I've been developing, a new mesocosm that has some of the advantages of the Soay sheep, but also allows me to experiment. So basically what we're doing is what we call experimental immunoepidemiology, and this is where Jackie, Sarah, and Tina are the central team. So we have a mouse enclosure. This is it viewed from Google Earth. This is it viewed from Martin Wakelski in a hang glider. He and Ayla Howe, about 10 years ago, set this up for studies of behavioral ecology, in fact, studying circadian clock biology in mice. It's 8 replicate enclosures, each of which has a little barn and feeders and this sort of thing. What we've been doing the last few years is adapting this for use in immunoparasitology. So we're basically taking mice out of their little clean box with all the wood chips and moving them into a farm. We are not using albinos for obvious reasons. We are introducing the nematode into the system, it's Trichuris murus, that Richard is the king of and gave an introduction, a beautiful introduction to the biology of that species. So we really are taking baby steps. This is actually a fairly big undertaking, and I'll tell you about our baby steps and then our longer term hopes and visions. We've started by asking, what is the contribution of environment to resistance? If we take super uber resistant lab mice out of that clean box and let them experience the natural world for a little while, does the resistance phenotype carry? And then returning to this resource cost question, can we detect that resistance comes at any sort of resource cost? So the things that I think this system shares with the soy sheep and what I would answer to the question of why study nematodes in mesocosm mice is that it enables us to study individual mammals longitudinally. We know their genotype if we're using inbred strains or even collaborative cross, advanced collaborative cross genotypes. We can also characterize the environmental variation well, and to a large extent, we can actually manipulate it within the confines of these replicate farms we have. So the first thing we want to do was test, as I said, whether resistance carried over from lab into field, when instead of living in wood chips with chow hanging right over their noses and drinking water in a bottle, they are burrowing like this. They're exquisite burrowers, it turns out, and drinking dew and otherwise living in this sort of semi-natural environment. You'll notice lots and lots of pie plates because we're, again, trying to exclude predators from the system. This is a review that Richard published last year on the cytokine biology of resistance against these nematodes. So I don't need to go into this in detail after his talk, but just wanted to say that the C57 black 6 mouse genotype that was one of the ones we released into these enclosures, they do lots of the T helper type 2 immune response, lots of the interleukin 13, and that is associated with resistance against these nematodes. We also released genetically modified C57 black 6 mice into these enclosures. We had these mice that are called STAT6 knockout, which basically turns off the Th2 immune response. So they had very little interleukin 13 and lots of interferon gamma. We sort of use this as a benchmark against which to measure susceptibility changes if we saw them. So just to give you a sense of this initial experimental design we've undertaken, we had 3 sort of environmental treatment groups. We had groups that never left the lab, maintained under traditional lab conditions, except we corrected for abiotic factors. We matched the photoperiod to the current New Jersey photoperiod. We matched temperature and humidity as well. But otherwise, they stayed in the lab the whole time. These guys went out into the field after we'd infected them with worms, and then they spent a couple weeks out in the field. And then these guys, we put out right at the start, and then infected them once we had trapped them after they'd been out in the field a while, and then they stayed in the field the whole time. And then we observed their immune and parasitological parameters at the end of this period. And it is a very short-term experiment. As I said, these are baby steps. But in the first instance, it was only up to week 4 post-infection. And we put both the knockout and the wild-type strains into these conditions. We only were able to put the wild-type mice into the long-term environment group. Okay, so what did we find? So we found, like some of the other tales we've heard this morning, unsurprisingly, but C57 black 6 mice do not seem to be quite so resistant anymore when they've been outside, even for a couple of weeks. So this is a plot of worm burden, comparing the mice that never left, the mice that spent a couple weeks in the field, and those that spent 5 weeks in the field. There's a lot of variation, negative binomially distributed for sure. And accounting for that, though, both of these are higher worm burdens at this time point than in the lab mice. And this is using what Richard was calling the high dose. So the benchmark, the STAT6 knockout mice, we kind of, when we released them, we were like, good luck, guys. We weren't sure how they would fare. But they seemed to do very well. They seemed to enjoy the life in the wild as much as the others. And sure enough, they were remaining very susceptible to worms. But in fact, statistically, there's no difference amongst these three groups. With 2 weeks of exposure in the field, the resistant genotype was no longer distinguishable from a genetically knocked out mouse. And the worms seemed to be super happy. They were big and strong, 10 times the biomass in the field mice compared to lab mice. So I would embrace mechanisms such as Richard explained about the interactions with the colonic microbes explaining this. Maybe the hatching success is that much higher when they've got a little bit of gut diversity in their microbial communities. Maybe the worms are feasting on all this extra microbial diversity. I don't know. Richard and I will have to put our heads together. But we also did look at the immunology, and this is just showing a representative plot. It's showing an interleukin-13 measured in the lamina propria cells, so T helper cells that are in the lamina propria behind the gut wall, proportion of the T helper cells in that tissue producing interleukin-13, and then in uninfected as well in infected mice. And in our lab mice did what they're known to do when they're infected. They make a lot of interleukin-13, and this is probably the proximate cause of their resistance. But this disappeared when they were in the field. And you'll notice that it's not just that the infected mice had less IL-13 going on. The uninfected mice, who didn't have the nematodes that we'd given them, there's other stuff going on. There's more IL-13 background in those groups of mice. We have by no means unpacked that yet, but there's certainly a location by infection interaction. So the last piece of the story, and this is very hot off the press, and I apologize for the rawness of these data, is that when we measure leptin, which is a fat-regulating hormone, in the mice in whom we've made all these other measurements, and look at the leptin levels compared to the interleukin-13, whether in the lamina propria or in the mesenteric lymph nodes, the production of that resistant cytokine. And then we also look at it in light of the worm biomass. We think we see the following, but I'd love to hear your thoughts on this plot. So first of all, the mice that we did not give worms, these open triangles, we do see that some of them are making quite a lot of IL-13, and our eyes are caught by the fact that the ones that are making a lot of IL-13 have pretty low leptin, which suggests low fat reserves, suggesting potentially a cost to that response. The pattern is more complex with the worm biomass, but I wanted to say that these guys, these are the mice that were in our infected groups, and I should say all of these are mice from the field. Mice in the lab can get up more into this right corner of the plot. These are all field mice. But these are the ones who did manage to clear their worms in the field. So these are the ones who had really low burdens. And it's interesting that they had very strong interleukin-13 responses, and their leptin was rebounding, suggesting potentially that the worms are gone, and then the mice can rebound from that. But we're wondering whether there is some sort of fat or at least a resource cost to this resistance that becomes unmasked in the field, and we're detecting it by looking at the relationship between IL-13 and leptin so far. Okay, so our first step was this sort of genotype by environment study. We do find that resistance seems to be labile by environment. The exact cause of that, I mean, there are so many axes of difference between the mice in the box and the mice out in these enclosures. We may or may not actually answer that, but we have quite a few angles at work. So please watch this space, and we will try to get to the bottom of that. But what we plan to do now is stop the lab versus field comparison and just study the field situation, but begin to manipulate the resistance by potentially manipulating the relative costs of parasitism and defense borne by these hosts. But when I say cost of parasitism, we can mess with the dose and ask hosts to cope with smaller or larger numbers of worms. We're also able to mess with resource availability, and what we've done so far is manipulating the nitrogen to carbon ratios of their diets. We're also cognizant that we get into a wide world of behavioral ecology here, and we're embracing this and learning how to proceed, but we realize we need to understand compensatory feeding and diversity in feeding habits, so we've very much undertaken that. And the aim is ultimately to really make these mesocosms in which mice can live out their lives. We're not there yet. So my final comment is that I'm hoping that with the diversity of different approaches to crossing this ecology to immunology divide that we've heard about in this symposium, including mice who are out exploring the world for the first time in their lives. I think our work and all the other we've heard about, I hope will lead us to a point where the hyphen will become so short that the whole word ecology can actually disappear, because we're all just immunologists. And I think we need each other from the mechanistic end through to the very wild end to really understand immune function. Okay, thank you.
Fantastic, really nice end to the symposium. I think if Andrea's voice will hold out, you can ask a question, but of course, if anyone wants to go for lunch, please go for lunch.
Well, we actually have all those other axes, and so far leptin captures that. But our guys are, of course, more standardized than yours, right? Ours have only been outside for 2 to 5 weeks. But yeah, it's a fair point, and we've got all the bits weighed, including the spleens. So I'll get back to you on the spleen story. Yes. Did people hear the question? Wondering what else they're exposed to when they're out there. So we don't know for sure, but we have done some fairly thorough searching. We look in the guts as we're looking in the guts for everything else. We've done 16S sequencing to look for the microbes, including any potential pathogens. And then we've done some serology. And as far as we can tell, one mouse got a pinworm, and nobody else got anything that is known. So again, it may be that we just need that when we have them out there for life, we'll really have to think harder about that. But oh no, somebody got a botfly as well. We had somebody with a botfly, somebody with a pinworm, but otherwise pretty depauperate. But we have to keep an eye on that in the longer term.